My name is Alexandru and I'm a second year cellular and molecular biology student at the University of Glasgow. This summer, I've worked on developing a curated database and a workflow for searching novel and existing bacteriocins produced by plant pathogenic bacteria. Bacteriocins are narrow spectrum, proteinaceous antibiotics that are present in many types of bacteria which target and kill related bacterial species. Therefore, they might become a better alternative to conventional antibiotics as they do not impact the environment or wider soil microbial communities and can be even expressed in plants or used as prophylactic treatment. I've applied for the project as my university advisor, Dr. Joel Milner, is actively involved in this area of research. Last year, we discussed about a potential summer internship in his lab, but due to the coronavirus pandemic, the plan was cancelled. So I'm glad that this year he reached out to me in order to take up this remote bioinformatics focused project. My project was integrated into a much larger multi-center project called DSBL, or Building a Decision Support Tool for Potato Blackleg Disease, which is a collaborative grant led by Professor Ian Toth at the James Hutton Institute. Therefore, the database and workflow were particularly used to look for bacteriocin signs in Pectobacterium atrocepticum genomes, as this is the main causative agent of potato blackleg disease in the UK, a disease which costs potato growers approximately £50 million per year. At the start of my project, I built a database of known bacteriocin sequences. This was done by mining existing databases found in literature, both manually and by using bash scripts. After the database was built, I started developing the workflow, which would take bacterial genomes as input, with the output being multiple visualizations and proteins that match bacteriocins. The workflow is comprised of genome annotation, pangenome analysis, protein alignment, and multiple visualization tools. Is written in Bash, R, and Python and was a lot of fun to work on. I then tested the workflow on all publicly available Pectobacterium atrocepticum genomes, plus 8 more genomes which came from the James Hutton Institute. I obtained hits which resemble bacteriocins, which were further characterized by multiple sequence alignment and phylogenetic analysis. I absolutely loved the project, and I've learned a whole lot about bioinformatics, bacteriocins, and Pectobacterium control. I also had the opportunity to work on my presentation skills, as I would participate in weekly progress meetings with the other university staff working on the TSBL project. Working on this project made me realize just how much these two fields, bioinformatics and plant pathology, can work with each other, and I would recommend everyone who has a passion for both to apply for a BSPP studentship. Last but not least, I would like to thank the BSPP for this opportunity, as I believe that this project acted as the foundation towards a future PhD and research career. Thank you.